I work on E. coli, which is the superhero of the pharmaceutical industry. It often acts as a workhorse in many different processes, from producing insulin to antibiotics to anti-cancer drugs. However, it's also a villain, with one particular nasty strain of E. coli affecting over 250,000 people alone in the United States last year. These villains often have an arsenal of weaponry, including toxins and antibiotic resistance. What often turns these superheroes into these villains are proteins. These proteins are like puppeteers. They are controllers and can pull the strings within the bacteria, switching these weapons on and off. They do this by attaching to the bacteria's DNA and altering it. This is a really interesting subject to look at because if we can understand the bacterial DNA protein interactions, then we can start understanding the disease better, and this can hopefully start contributing to some therapeutic methods. My work primarily revolves around a single protein that is involved in controlling E. coli's ability to live within hostile environments in the body. Proteins can often control just one region of DNA, or they control multiple regions of DNA, and these are termed master controllers. My research therefore wanted to know, is my protein a master controller? What regions of DNA it controls? And are these affected in virulence? To do this, I first mapped out where my protein binds to my DNA. This allowed me to see where on E. coli's DNA my protein specifically bound to, and whether these regions were involved in virulence. This reveals some 80 new ways in which my protein binds to my DNA, altering it, suggesting it is a master regulator, pulling the strings in making E. coli harmful. I'm now starting to verify each of these results as well. This is a really interesting topic to look at because it's given us a clearer understanding of bacterial protein DNA interactions, as well as highlighting how just a single protein can have such a large effect on multiple processes within the bacteria, really showing how it pulls the strings in making the bacteria be harmful. But it's also interesting at the moment because it's given us targets that we can focus on in these disease-producing bacteria, which is really important in this era of antimicrobial resistance.